The project started in one of the most beautiful way, I think, one of the most beautiful way to make science, which is serendipity. One day, Mixum Falovecki, a brilliant PhD student at Nottingham, Trent University, noticed this beautiful pattern right here is one of the flasks where it was cultivating cyanobacteria. This beautiful, almost hexagonal pattern of the green filaments came to us and says, "Hey guys, can you help us explain this strikingly geometrical pattern which we do not expect, in this colony of cyanobacteria?"
So these are the organisms which basically invented photosynthesis. They oxygenated our atmosphere. Before them, there was no oxygen. It was just nitrogen, carbon dioxide, so on. And even now, they're responsible for a great amount of the oxygenation of our planets, the majority of the nitrogen fixation in our seas. This algae grows everywhere. So it is something which is covering our planet, but we don't even know how exactly it moves. So these cells stick together in long lines, long chains. These chains then bundle together into ropes, filaments, and these filaments then grow into larger structures. These structures are obviously important. They allow the algae to have some common defense against the environment. They give it some sort of a rigidity, resistance to turbulence, motion, flow. But even the basic questions of how these algae move or how they interact with each other or how these structures arise have been absent.
We devised a theoretical model to understand the motion of these cyanobacteria, and then we worked in close collaboration with our experimental colleagues so that we would capture the real behavior, the real phenomenology of the system. We found that a very simple principle, the fluctuation in the shape of the filaments actually set the length scale of the pattern, the hexagonal pattern that Mixon one day, some years ago, showed us in his flask, in his cultivation flask.